Hello my dear students, a very good afternoon to all of you, and I welcome you all to the most interesting and interactive live session. This is Vasavi Ma'am, your biology educator for Foundation and CBSE. Yes, students, in the yesterday's live session class we have discussed about the discoveries of cell, and we have also studied in detail about the composition of the cell based on the number, size and its shape. Okay, and later on we have also discussed about the types of microscope. Okay, and we have learned about the compound microscope as well as the electron microscope. Okay, and in today's particular session we are going to discuss about the differences between the prokaryotic cell and the next eukaryotic cell. Yes, students, yesterday I have gave you one question that what is going to form the major difference, what is the major difference between the prokaryotic cell and eukaryotic cell. So here comes the answer. Yes, in the eukaryotic cell, we have given the term called true nucleus. Okay, in the eukaryotic cell what is present, children? True nucleus is present in the eukaryotic cell. So the true nucleus is going to be present and it is going to possess the important characteristics like nuclear envelope. Yes, this already we have discussed yesterday. Okay, so let's recall it once again. Yes, nuclear envelope, nucleoplasm, and the next one nucleolus. Okay, these are all the characteristics which we can find in the eukaryotic cells, and in the eukaryotic cells the genetic material is present inside the nuclear envelope. Okay, the genetic material is entirely covered by the nuclear envelope. Okay, so now along with this genetic material which is surrounded with the nuclear envelope, it is also going to contain some many different types of membrane bound cell organelles like mitochondria, Golgi body, endoplasmic reticulum, and the next lysosomes, cytoplasm, and the next ribosomes. These are all the complexity structures which we can observe in the eukaryotic cell. Yes, now keeping these concepts in mind, okay, let's now try to understand in detail about what is prokaryotic cell. Okay, clear children? Yes, this is the structure of the prokaryotic cell and it is a bacterial cell. Yes, these are the microscopic structures of the bacteria. So these bacteria is made up of only single cell. It is a unicellular microorganism. So now we have to study about this single cell. Okay, so when we study in detail about this single cell, it is going to actually look in this manner. Okay, and it is going to be composed of many different types of parts like nucleoid region, pilus, ribosomes, food granule. We are going to discuss it now. Yes, children. Okay, yes. So this is the ultra structure of the bacterial cell. We can also call it as a typical structure of the prokaryotic cell. So now this prokaryotic cell is going to possess three important characteristic features. Those are, yes, this bacterial cell is going to possess three most important characteristics. Those are glycocalyx, and the next cell wall, and the next one is cell membrane. Clear? Yes, these three are the most important characteristics which we can see in the prokaryotic cell, whereas in the eukaryotic cell there is only a nuclear envelope which is going to be surrounded around the that genetic material. But here when we are going to see, it is going to contain the glycocalyx, cell wall and cell membrane. So now let's discuss about each and every structure in detail. So now first we are going to learn about the glycocalyx. So children, now generally every cell is going to be surrounded by the cell wall and the cell membrane along with the cell wall. Okay. So now, yes, in the bacterial cell, that cell wall is again additionally surrounded with some most important protective layer that is called as glycocalyx. Okay, glycocalyx is a extra protective layer which is going to be present around the cell wall. Okay, around the cell wall again one additional layer is present which gives protection to the bacterial cell, that is called as glycocalyx. Okay, yes. So now here if we look into this diagram, if you look into this structure, yes, this is going to form the glycocalyx. Clear children? This is the glycocalyx. Which one? This one, this portion. Okay, this is the glycocalyx, and this glycocalyx is made up of two components. Glycocalyx is made up of two components. Yes. Okay, this glycocalyx is made up of two components. One is slime layer and the next one is it is also called as mucilage layer or capsule. The slime layer is made up of proteins and mucilage layer is made up of, the capsule is made up of what children? The capsule is made up of carbohydrates. Okay, this capsule is going to secrete the mucilage substance and it is going to prevent the glycocalyx structure and it is going to prevent the bacterial cell from the dehydration. Okay, from the loss of water, that capsule is going to prevent the glycocalyx from dehydration process, because this capsule itself is only going to secrete the mucilage. Okay, yes. And the next one is a slime layer. Slime layer and the capsule both are sticky in nature. So, these both are together called as the glycocalyx. This slime layer and capsule will help in attachment of the bacterial cell to the host. Okay, it means this glycocalyx is going to be capable of digesting from the host enzymatic activities. Suppose if I am the host, okay, so suppose if I am the host and in my body if there is a bacterial cell, so in our body many enzymatic reactions are going to take place in our daily life. Okay, many enzymatic reactions will occur and HCl is also going to be released during the digestion process. So at that time if there is a bacteria in our body, so what makes the bacteria, okay, what makes that bacterial cell to survive in our body? Okay, this is a question to you children. Okay, so what is going to make, okay, what is going to make that bacterial cell to survive in our body? Okay, it's the answer will be absolutely glycocalyx. Clear children? So the glycocalyx is an extra protective layer which is going to be present around this bacterial cell. Okay, it will help in attaching to the host body, and even though if we are going to release many enzymatic reactions and even though if we are going to release acids in our body, that glycocalyx will help in protection. It is going to give the additional protection to that bacterial cell, and that glycocalyx will also going to help in preventing the bacterial cell from the dehydration. Dehydration means loss of water. Okay, it will help in preventing the drying. Okay, it actually prevents the bacterial cell from the drying, because actually the capsule itself will release the mucilage substance which will keep the moist structure to that glycocalyx to attach the host host body. Okay, clear with the glycocalyx? Yes, and it is also going to show the immunogenicity children. Okay, what is immunogenicity? So immunogenicity is nothing but just identifying that bacterial cell as a foreign material. Okay, as I have said that if there is a bacteria in our body, if we identify that bacterial cell as a foreign substance or any foreign material, that identification is only called as the immunogenicity. Okay, and sometimes if we want to kill that bacterial cell, if we want to kill this bacterial cell in our body, even if we are going to take many antibiotics also, sometimes the bacterial cell will never gets broken down. Why? Means there is a glycocalyx which will give the protection to that bacterial cell. So in that cases, even though if we are not able to recognize that particular cell, even though if we are not going to recognize that particular bacterial cell in our body, at that time what happens children? It is going to show the process called as virulence. Okay, virulence means incapable to identify that bacterial cell as a foreign substance. Are you getting my point? Clear? Virulence is nothing but we are not able to identify. When we are unable to identify that bacterial cell as a foreign material, then it is going to cause disease in our body. That is only called as virulence. Okay. and immunogenicity means identifying this bacterial cell as a foreign substance. Yes. And that glycocalyx is also involved in what? It is also involved in preventing the bacterial cell from desiccation. It means it prevents that cell from drying. Okay. These three are the most important functions which is going to be performed by the glycocalyx, which is surrounded around this cell wall. Around the cell wall, this glycocalyx is going to be surrounded, and this glycocalyx will help in showing all these activities like virulence, immunogenicity, and the next desiccation. It is actually composed of slime layer and the next capsule, which is made up of proteins as well as carbohydrates. Yes. Clear, children? Yes. Now we are going to look into the internal structure of this bacterial cell. If we look into this internal structure, yes, inside this bacterial cell there is a fluid filled substance. Okay, there is a fluid filled substance that is called as cytoplasm. Okay, inside this bacterial cell we have the cytoplasm. So here is an important point, so everyone have to try to understand this. Yes, inside this cytoplasm we have the genetic material. Okay, we have the genetic material inside the cytoplasm. Okay, so now just let me explain you the general structure of a cell. Yes, so generally if we see, this will be the cell. Okay, inside the cell we are going to have the cytoplasm. Yes, this will be the cytoplasm which is going to be present, which is the fluid which is present in the cell. Okay, yes, cytoplasm, and inside this cytoplasm, yes, let me fill up this, children. Okay, clear? Yes. So inside the cytoplasm now we have the genetic material. Children, now we are discussing this regarding with the prokaryotic cell. Okay, don't try to forget it. Yes. So here we have the genetic material. Okay, this is all the circular genetic material which is present inside the cell of the prokaryotic cell structure. Okay. So now this genetic material is not going to be surrounded by the nuclear membrane. Okay, nuclear envelope is completely absent in the bacterial cell. Nuclear envelope is completely absent in the prokaryotic cell. Hence here we are going to call it as a primitive type of nucleus. Okay, or else we can also call it as an incipient nucleus. Okay, so here it is not going to be covered by any nuclear envelope. So here completely nuclear envelope is absent. Okay, so that nuclear envelope is going to give the definition for both the prokaryotic cell as well as eukaryotic cell. If the genetic material is going to be covered along with the nuclear envelope, then we call it as a true nucleus, which is going to contain the nucleolus and nucleoplasm, that we can see in the eukaryotic cells. But coming towards these prokaryotic cells, the genetic material is freely floating in the cytoplasm without covering with any nuclear envelope. Okay, nuclear envelope is absent. Okay, so it is just freely floating in the cytoplasm which is present in the prokaryotic cell. So along with this genetic material, okay, there in the eukaryotic cells we have named it as a true nucleus, but here when it comes to the prokaryotic cell, we are calling this genetic material as nucleoid. Clear, children? So this genetic material which floats freely in the cytoplasm of the prokaryotic cell, that genetic material is called as nucleoid. Okay, it is very, very important. Okay, so based on this concept only, now we are going to solve the 4 marks question. Okay, that is a long question, so you have to understand this concept clearly. By that you can attempt the answer, children. Okay, so here this is called as nucleoid. Okay, it is called as nucleoid, whereas it is called as nucleus in the eukaryotic cells. Yes, along with these in the cytoplasm we are going to find the non-membrane bound cellular organelle structures. Those are called as ribosomes. Can you see these black color dotted structures? These are actually the ribosomes which are non-membrane bound. Okay, these are actually meant for protein synthesis. Okay, yes. And inside this cytoplasm we have the food granules. Food granules are present inside the cytoplasm. These are meant for they can store the different types of foods like glycogen. Yes. Okay. And then next, inside the cytoplasm only we are going to have the gas vacuoles, children. Okay. Inside the cytoplasm we have the gas vacuoles. It is not visible, I think. Yes. Okay, we have the gas vacuoles. Now what is the function of that gas vacuoles? The gas vacuoles are going to be present in the bacteria. That gas vacuoles will help that bacteria to float on the surface of the water. Okay, they are going to create a nature called as buoyancy. Okay. So, buoyancy is nothing but they are the floating nature of the bacterial cells. Due to the presence of the gas vacuoles, they can easily float on the surface of the water. Okay. And next, we also have the photosynthetic pigments in these bacterial cells. Those are called as, yes, photosynthetic pigments are also present in the bacterial cells. Yes. Okay. And then next, yes, along with these are completely located in the internal structure of these bacterial cells. Okay, inside the structure what we have the free floating genetic material which is called as the nucleoid, and then next we have the ribosomes. Along with this we have the food granules which will store the glycogen, and next gas vacuoles, and then next one some photosynthetic pigments. Okay, and these gas vacuoles will help in floating the bacterial cells along the surface of the water. Okay, now if you look into the external structure, okay, yes, below the glycocalyx what we have? Below the glycocalyx we have the cell wall. Okay, below the glycocalyx we can find the cell wall, children. Yes. So now cell wall of the bacteria is made up of peptidoglycan. Peptidoglycan. Okay. So children, in your NCERT standard textbook only the structure of the prokaryotic cell is given. So I request each and everyone to note down all these points as they are going to be very important in your higher standards also. Okay. So the cell wall of the bacteria is made up of peptidoglycan. Okay. Yes. And this peptidoglycan is going to be arranged towards this entire, yes, it is going to be surrounded towards this entire prokaryotic cell. Okay. This peptidoglycan is made up of proteins and carbohydrates. Clear, children? Yes, peptidoglycan is a cell wall which is present in the prokaryotic cell. It is made up of the repetitive units of the proteins as well as the carbohydrates. So now cell wall of the fungi is made up of chitin. Cell wall of fungi is made up of chitin, and then next cell wall of plants is made up of cellulose. Now next we are going to study about this cell wall in detail. So I am just explaining you in a brief. Okay. So the cell wall of the plants is made up of cellulose, and cell wall of fungi is made up of chitin, and the next cell wall of the bacteria is made up of peptidoglycan. These three are very important. Okay, these three the cell wall structures are very important for your examination point of view, as well as they are also going to form the questions in the section A, children. Okay, yes, clear about the cell wall? Cell wall is made up of peptidoglycan. Yes, below the cell wall we have plasma membrane. Okay, each and every cell is going to have plasma membrane, and that plasma membrane is surrounded from the external surface with the cell wall. So now this plasma membrane is made up of phospholipid. Plasma membrane is made up of what, children? Phospholipids. Okay. Now we are going to learn about this plasma membrane and cell wall in detail. Okay. So here just you need to understand its outlook. Clear about this structure, clear about the glycocalyx and its functions, and next what we have discussed, cell wall and then next plasma membrane, and coming towards this internal structure we have discussed about this genetic material which floats freely in the cytoplasm, that is called as the nucleoid. So here one more interesting thing you have to remember. So this is called as the plasmid, children. Okay. So plasmid is the extra chromosomal circular DNA. Okay. Yes. About the plasmid also you are going to learn in your higher standards. So it will be better if you are going to learn it in short form. Okay. So there is an extracircular chromosomal DNA that is called as plasmid. Okay. So the plasmid is an extra chromosomal circular DNA. It can replicate itself without depending upon this nucleoid. Okay. It can replicate and it can survive by its own without depending upon its nucleoid. Okay. And next, if we look into the external projections, if we see the external projections arising from this bacterial cell, we have pilus. Okay. Yes. Pili, and the next one we have the flagella. Yes. These small hair-like structures. Okay. The whatever the small pointed hair-like structures you can see around on the outside surface of this bacterial cell, these are called as the pilus. So here comes the most important function of this pilus. So during the sexual reproduction, these pilus are going to create the conjugation tubes. Clear, children? Yes. They will create the conjugation tubes. So from these conjugation tubes, the genetic material can be passed from the one bacteria to the another bacteria. Okay. And next we have the flagella. Okay. This is a flagella which is going to be connected to the intercellular structure. Okay. This flagella is completely embedded inside this intracellular structure, and now the function of the flagella is to give the movement to this bacterial cell. It is going to help in the locomotion, children. Okay. Flagella is just meant for the locomotory organelle, which will show the movement, which will help the bacterial cell to move from one place to the another place. And this flagella is made up of a protein called as flagellin. Flagellin is a protein which is going to be composed, which will give the structure flagella. Okay. And it is connected to this intercellular structure with basal body. Hook, and yes, this will be the basal body. Yes, and then next this will be the flagella. Clear, children? Clear with the concept prokaryotic cell? Yes, in the prokaryotic cell, the entire bacterial cell is going to be covered with an extra protective layer that is called as the glycocalyx, and that glycocalyx is made up of slime layer and capsule. This will help the bacteria to attach to the host body. So this glycocalyx is going to give extra protection to the bacterial cell from the host enzymatic activities. Okay. So the glycocalyx is going to perform the three most important functions. Okay. It will give protection to the bacterial cell, it will prevent the bacterial cell from the desiccation, it is also going to show the immunogenicity, and the next virulence. Okay. And next, coming towards the cell wall, it is made up of peptidoglycan, which is composed of both proteins and carbohydrates. Okay. So continuously it is going to be arranged towards the entire bacterial cell. And if you look into the intracellular structure, we have the cytoplasm where the genetic material floats freely without covering with any nuclear envelope. Okay. This nuclear envelope is going to give the proper definition to both the prokaryotic cell as well as eukaryotic cell. And along with this, here we have the ribosomes. These are non-membrane bound cell organelles which are involved in synthesizing the proteins, and along with this we have the food granules which will store some food material in the form of glycogen. And next, here we have the extra chromosomal circular DNA. Okay. It is involved in biotechnological process. Okay. As if now it is not required. So if you remember, it is very good to learn extra also, children. Okay. And if we see the external projections, there are some hair-like projections which are going to arise from this bacterial cell. Those are called as pili. Okay. And here we have the flagella which will give the bacteria cell and it will show the locomotion. Okay. And flagella is made up of a protein called as flagellin. Okay. Clear with the concept prokaryotic cell and eukaryotic cell? Yes. Now, yes. So this is an NCERT structure. Okay. Yes. just now whatever the concept we have discussed, these are the important points from that. Yes, so it is going to surround the cell wall of the bacteria and it is made up of slime layer and capsule. Yes, we have already discussed the functions which will prevent it from desiccation, and next it is going to protect from the host enzymatic activities, and along with this it will show the immunogenicity and virulence. Yes. Yes, children, just a minute. Yes. Yes, this is a most very important for your examination point of view. It is going to come for 5 marks or 4 marks, children. Okay, the differences between the prokaryotic cell and eukaryotic cell. So now by looking into these diagrammatic structures, you need to answer these particular concept, children. Okay. So if we look into the first point in the prokaryotic cell, cell wall, if present, it is composed of peptidoglycan. Okay. As already I have informed you just now, the cell wall present in the bacteria is called peptidoglycan. Yes, done. And next in the eukaryotic cells, yes, in the plants also we have discussed about the cell wall. The cell wall in the plants is called as cellulose. Okay. The cell wall in the plants is made up of what, children? Cellulose. Okay. And next point, a typical nucleus is absent. Why a typical nucleus is absent in the prokaryotic cells? Yes, in the prokaryotic cells, free genetic material, free nucleoid is present, which is not going to contain the nucleoplasm, nucleolus and the next nuclear envelope. All these three structures are absent in the prokaryotic cell. Okay, so hence we are calling it as a typical nucleus is absent. Okay. But whereas coming towards the eukaryotic cell, typical nucleus is going to be present. And here DNA is located freely in the cytoplasm. Okay. See here, nucleoid, it is located freely in the cytoplasm. But here, yes, DNA is located inside the nucleus. Yes, this is a nucleus and here we can find the genetic material, and along with this nucleus, the mitochondria and chloroplast are also going to have their own genetic material. Okay. And next here DNA is circular, where in the prokaryotic cells DNA is circular. Okay. Whereas coming towards the eukaryotic cells, the DNA is linear. Okay. This is a very important concept. So I request each and everyone, so you can take the screenshot of this particular topic. Yes. Yes, children. Yes. Now coming towards the subtopic cell wall. Yes. Next coming towards the cell wall. Yes. So in the plants, cell wall is going to be made up of primary cell wall and the next secondary cell wall. Okay. So first we are going to have the plant cell. Okay. The plant cell is surrounded by primary, first from the outer to inner it will have the secondary cell wall and the next one primary cell wall. So this primary cell wall is cellulose. Okay. Primary cell wall is made up of cellulose. It is a porous and permeable. Permeable in the sense it can allow the molecules in and out. Okay. And it is permeable in nature. Below the primary cell wall we have the secondary cell wall, which is made up of lignin and suberin. Yes, lignin and suberin, whereas it is impermeable in nature. Okay, we are talking the cell wall structure in based on the plant cell. Okay, in the plant cells, primary cell wall is made up of cellulose, which is permeable in nature, and secondary cell wall is made up of lignin and suberin, which is impermeable in nature. Okay. Yes. And next in the bacteria, the cell wall is made up of peptidoglycan. Yes. And in the plants it is made up of cellulose, and in fungi it is made up of chitin. Yes. This is one small subtopic, children. This is also important, but here you need to remember only these types. Whether in bacteria, what type of cell wall, when it comes to the plants you need to mention its cell wall, as well as fungi along with its cell wall. These three are most important. Yes. So children from yes. So up to now we have completed some most important topics which are important for your examination point of view. So based on that all the analysis, here are the few NCERT exemplar questions which are already included in your NCERT's 9th standard book. Okay, so please everyone try to answer this. Yes. Name the smallest sized organism. Okay, yesterday only we have discussed about that smallest living organisms. This question is based on what, children? It is based on the composition of the cell. The composition of the cell includes cells based on number, cells based on the size, cells based on the shape. So here the smallest sized organism, okay, name the smallest sized organism. The smallest sized organism is mycoplasma, okay, it is also called as pleuropneumonia like organisms, okay. Now coming towards the size, it is going to range from 0.1 to 0.5 micrometers. It is very, very important for 1 mark. So please everyone try to note down this. Yes. And next, who discovered cells and how? Okay, this question this question is related to discoveries of cells. Okay, we have already discussed it in detail. Who discovered the cell? Robert Hooke discovered the cell in the year 1665. Okay, so he studied the, yes, where he how he discovered the cell? He examined the slices of cork under a self-designed primitive microscope. Okay. So when he observed that cork structure, it is going to resemble the structure of honeycomb. Okay. If you look into the previous class, there everything in a brief we have discussed it. So you can answer it carefully when you are going to be thorough with the all the sessions, children. Yes, honeycomb-like structures. Later, then next he consisted that many tiny compartments. Okay, and next they called them as cellulae, and now they are going to term as cells. Okay, cellula is a Latin word which means hollow structure. Okay, when Robert Hooke studied all the when Robert Hooke studied this cork structure under the microscopes, there he observed the some compartment like structures which are going to resemble the honeycomb like structures. Okay, so that honeycomb like structures he called it as cellula, which means those are going to give the term cells. Okay. Yes, children. Yes, coming towards the next question. Why is the cell called the structural and functional unit of life? Why cell is called as the structural and functional unit of life? Yes, see here, all the living organisms are made up of cells. Yes, we have already discussed about it also. And next, the cell is the structural unit of life. Each cell acquires a distinct structure and function. Cell gives structure to the organism as well as it is also involved in functions, and we have also seen the example of unicellular and multicellulars. Okay, in the unicellular like amoeba, different types of functions is carried by only single cell. That is the reason it is called as the functional unit. And in multicellular organisms, group of cells will form the tissues and the next later organs and the next organ systems and then next later it is going to form the complex individual called as organism. Okay. So, in the multicellular organisms, due to this complexity arrangement, it is going to give the proper structure to the organism. Hence, it is also called as the structural and functional unit of life. These three are very, very important answers for both 1 mark, 2 mark as well as 3 marks. They are going to form short answers as well as very short answers. Yes. I hope everyone are clear with these all the concepts. So, now here comes the most important and very, very interesting topic, that is plasma membrane structure. Yes, plasma membrane structure. So, children, from this particular topic plasma membrane, you are going to get case based questions. Okay, and then next you may also expect the 1 mark questions and then next very short answer type of questions. So, these three are the different categories of questions which you are going to expect from this particular chapter plasma membrane. Okay, it is a very interesting topic. It is the plasma membrane structure. It is very important as well as it is very interesting to learn, children. Okay, so I request each and everyone to be stay tuned until this plasma membrane structure and until this section gets end. Okay, it is a very important for your examination point of view. Yes, just a minute, children. Yes. So what is plasma membrane? Okay, so look here, it is a cell, right? It is a cell. The each and every living organism, that may be either prokaryotic cell or else that may be either eukaryotic cell. So each and every cell is going to possess the plasma membrane. Clear? So if you see the arrangement of the plasma membrane, yes, now we are going to see just the outlook, how actually the plasma membrane is going to look like. Yes. Okay, this is the cell. Cell is going to be surrounded by the plasma membrane. Okay. Yes. The plasma membrane is called as phospholipid bilayer. Okay, plasma membrane is called as the phospholipid bilayer, or else the plasma membrane is also called as the cell membrane. It is made up of the phospholipid bilayer. Yes. Clear, children? These are all the phospholipids which are arranged in 2 layers, hence that is the reason it is made up of phospholipid bilayer. Yes, now this is a cell which is going to be surrounded by the cell membrane, and outside the cell membrane what we have? Cell wall. Okay, now we need to discuss only about this plasma membrane structure. So now coming towards this particular subtopic, so plasma membrane was discovered by the scientist called Singer and Nicholson. Singer and Nicholson discovered the plasma membrane structure in the year 1972. In the year 1972, Singer and Nicholson scientists proposed the plasma membrane structure. Okay, and they have by observing this structure they have proposed the plasma membrane and they have given it as fluid mosaic model. Fluid mosaic model. They have named it as a fluid mosaic model. Okay, this is a widely accepted model. So this plasma membrane is made up of the phospholipid bilayers. Yes. Yes, children. So this is the cell. Okay, so this is the green color structure, whatever I am drawing now. Yes, this is the intracellular fluid. This is intracellular fluid, and next here we have the extracellular fluid, ECF, extracellular fluid. Clear? And next these are this structure is plasma membrane. Clear, children? Every cell is going to be surrounded by the plasma membrane. Okay, plasma membrane is actually made up of phospholipid bilayers. Okay, the lipid layers are going to be arranged in 2 layers. Okay, so hence it is called as phospholipid bilayer. So here we have the intracellular fluid and here it is all the extracellular fluid, and in between here we have this plasma membrane. Okay, so generally what is the role of this plasma membrane in the cell? That plasma membrane is actually involved in transporting the ions from inner cell environment to the external cell environment. Okay, it is just actually meant to transport the ions and molecules. Okay, so based on this plasma membrane permeability, it is of different types. It is of different types. It may be impermeable in the sense it cannot allow the molecules, to enter the cell, as well as it cannot allow the molecules to move the ions or molecules from the cell environment to the outer cell environment. Okay, it is impermeable in the sense no permeability, no movement of molecules are going to occur. And next here permeable in the sense it can allow the solutes and solvent molecules from the external cell environment to the internal cell environment. And next, semi-permeable in the sense, semi-permeable means only few solvent molecules can enter into the cell. And next here, selectively permeable in the sense, only selected solute molecules can enter into the cell and only selected molecules can travel outside of the cell. Clear, clear with the concept? Impermeable, permeable, semi-permeable and selectively permeable. So now this plasma membrane is selectively permeable in nature. Clear children? Plasma membrane is selectively permeable in nature. It can select only the particular solute molecules or ions which are going to enter into the cellular environment, and impermeable in the sense which cannot allow the molecules to enter, permeable in the sense both solutes and solvents can enter, and semi-permeable means only some solvent molecules can enter into the intercellular fluid, and selectively permeable means some few selected solute molecules can enter from the outer cell environment to the inner cell environment. Yes, clear with the basic definition of the plasma membrane? Yes, now we will discuss how the molecules are going to transport from the outer cell environment to the internal cell environment. Yes, so for this you need to understand the general composition of the plasma membrane. Yes, so if you see the general plasma membrane composition, it is made up of phospholipid bilayer. Yes, and next these are tails. Clear children? This is actually just I am drawing the plasma membrane structure. Clear? So these are all the phospholipid bilayers. Yes, and here in between these we have the protein molecules. So in between the plasma membrane, what are going to be scattered? Some proteins are scattered in the plasma membrane. Okay. So in between these both the lipid bilayers, proteins are going to be embedded. So these proteins, the role of these proteins in the phospholipid bilayer in the plasma membrane is just to transport the ions from the external cell environment to the internal cell environment. Clear children? Yes. And the proteins are not only going to be present in the middle, there are some proteins which are going to be associated on the cell surface. Okay. So when proteins are present, when the proteins are going to be buried inside the phospholipid, they are called as intrinsic proteins. Intrinsic proteins, and the proteins which are associated along with the surface of this particular structure, these are called as extrinsic proteins. So now if we are going to study, if we see the single phospholipid, it is made up of, yes, it is made up of hydrophobic head, yes, hydrophilic head and hydrophobic tail. Yes, it's not visible, just a minute students. Yes, the single phospholipid, if we look into the single lipid structure, it is made up of the head. Okay, it is made up of hydrophilic head, and then next it is made up of the hydrophobic tail. Clear with the structure? This is the phospholipid bilayer. Here some few proteins are going to be scattered. These proteins are actually meant to take the molecules or ions from the external cell environment and they will transport it into the internal cellular environment. These are called as intrinsic proteins, whereas some proteins which are going to be present on the surface of the plasma membrane, those are called as the extrinsic proteins. So now we have to look into the lipid structure. Okay. So the single lipid structure is made up of hydrophilic head. Okay, it is made up of hydrophilic head, hydrophilic, and the next tail is called as the hydrophobic. Clear children? It is the head, this is called as the head part, I am writing it as a H, and this is called as the tail part. Head is hydrophilic in nature. Hydrophilic in nature means what children? Hydrophilic in the sense water loving. Okay, it is closely associated with the water molecules, as we have already seen in the introduction of the plasma membrane itself. The outside the cell we have the extracellular fluid, inside the cell we have the cytoplasm, and in between this we have what? Plasma membrane. Okay, so yes, once again I am showing you here. Yes, so these all will be these all will be the lipid structures. Okay, which are going to be surrounded with the cell. Okay, and here we have the intracellular fluid. Okay, and here we have the extracellular fluid. Okay, these head molecules, okay, these hydrophilic heads are closely associated with the extracellular fluid, as well as they are also closely associated with the intracellular fluid. In between these we have the hydrophobic tails. Clear? Clear with the concept? Okay, hydrophilic heads are associated with the water molecules. They are in close contact with the extracellular fluid as well as intracellular fluid. It means they are water loving. Okay, and here hydrophobic tails are present in between these heads. Okay, so hydrophobic in the sense water hating. Okay, so these from here it is going to start the transport mechanism. Okay, how the solvent molecules or solute molecules are going to be transported from the outer cell surface to the inter cell surface. So here comes a question for you that yes, what can you transport from this plasma membrane? Can water get transported from the plasma membrane? Can we transport the water across this plasma membrane, children? Okay, can we transport or not? Okay, to understand that you need to study the types of mechanisms. Okay, you need to study the transport mechanisms of the plasma membrane. Okay, the transport mechanisms are of 4 types. Yes, one is diffusion. Yes, diffusion, and next one osmosis, and next we have active transport, and then next later we have the bulk transport. Okay, these 4 are the transport mechanisms present in the plasma membrane through which you can transfer the molecules or ions from the extracellular environment to the intracellular environment. Okay, so I asked you a question that can water get transported from the plasma membrane. Okay, it is a question to all of you students. So please just to try to understand just whatever we have discussed about this structure now, please try to memorize it. So in the next session, in the tomorrow's live session, we are going to discuss these 4 transport mechanisms related to the plasma membrane. These are very important. Okay, so please stay tuned for the tomorrow session, and please like, share and subscribe to Neet World. Thank you.